This is lecture 16 on respiratory anatomy and gases. Over the next 3 lectures, we're actually going to be covering the respiratory system. Now, the functions of the respiratory system are as follows. First, it's going to be involved in gas exchange. Now, this is its primary function. It provides an area for gas exchange between the air and blood and is going to bring gases in and expel gases out. The respiratory system is also involved in the protection of itself. It protects itself from preventing surfaces from dehydrating, as well as preventing pathogens from coming into the actual respiratory system. It's going to be involved in the production of sound in verbal communication. It's what gives us voice. And finally, it is involved in regulation. The regulation of both the body fluid pH as well as water and heat loss. Now, what does respiration mean? Respiration in itself first refers to cellular respiration. Cellular respiration, as you remember, is the combination of oxygen and glucose in the cell to create ATP and CO2. Now, we get respiration at the cellular level from external and internal respiration in the respiratory system. External respiration is going to involve first ventilation, which we know of as breathing. It's moving air into and out of the lungs, and gas exchange. Gas exchange occurs between the lungs and the blood. The lungs bring in oxygen and exchange that oxygen with the blood. The blood will actually exchange and release CO2 into the lungs in order to exhale it. Internal respiration is going to be the transportation of gases in the blood, so moving the gases from the lungs throughout the body. And once we get to the different tissues of the body, this is where gas exchange will happen, again between the blood and the cells. From the blood, it will unload oxygen into the tissue, and the tissue will unload CO2 and put it back into the blood to be able to be breathed out in the lungs. Anatomically, the respiratory system is going to be divided into 2 branches. The upper respiratory system, which consists of the nose, nasal cavity, sinuses, and pharynx, and the lower respiratory system, which consists of the larynx, trachea, bronchi, bronchioles, and alveoli. You can notice the division happens specifically at the larynx. The nose, nasal cavity, sinuses, and pharynx are all involved in multiple systems, so not just respiratory system. The lower respiratory system is respiratory only. You can also divide the respiratory system based upon function. This is the conducting pathway versus the respiratory pathway. Now, the conducting pathway is where all air passages from the nasal or oral cavities through the terminal bronchioles of the lungs are conducting pathway. And then finally is the respiratory pathway. Now, this is where exchange happens. So it's only the respiratory bronchioles, alveolar ducts, and alveoli. Here we can see a nice diagram of the upper and lower respiratory system. And we can notice again the division between those 2 branches are going to be at the larynx, where it's going to actually just become respiratory only. Almost all of the respiratory system, except for the lower pharynx, terminal, and respiratory bronchioles in the alveoli, is going to consist of the following: pseudostratified ciliated columnar cells. This is going to make up a majority of the epithelial tissue that lines the respiratory system. The pseudostratified ciliated columnar cells have mucus-producing goblet cells. Your pharynx is going to consist of stratified squamous cells. Now, the large, thicker layers of epithelial cells are going to help prevent abrasion, and the ciliated pseudostratified columnar cells are going to help prevent dust and other pathogens from coming in and really kind of whisk it away with the cilia. As I've mentioned, a majority of the respiratory epithelium is the ciliated columnar cells. They produce mucus in an upward manner, and this is going to help it create a mucus escalator or elevator where it allows itself to eliminate debris that can be either coughed up or pushed back into the digestive tract for digestion. The mucus cells are going to produce this mucus, so inhaled debris will get stuck and not enter the lungs. And then finally, there are stratified squamous cells in the pharynx that provide protection against abrasion in food. Overexposure to foreign substances, things like bacteria or irritant chemicals, can cause an overproduction of mucus. This happens, of course, in a viral aspect, and we know this as a cold. Now, the upper respiratory system is going to be a pathway for air to go through. There's no exchange that happens. Air just simply comes in. Air will enter into the external nares. These are going to, of course, be the nose holes that you have. This passes by the nasal vestibula, where it has long hairs to trap the large particles or insects. Once it enters into the nasal cavity, it is separated into the left and right sides by the nasal septum. The anterior is hyaline cartilage. The posterior is a perpendicular plate of the ethmoid vomer. This is going to extend back and go into the nasopharynx. Here we can see the upper respiratory pathway, and we can actually see where the nose is, the nares, and then we can try and trace that down and back. So air would flow through the nose back to what we know as the nasopharynx, the back of the nose area. It would then extend back down to the oropharynx, which is going to be here, where the back of the oral cavity ends, and then down into the laryngopharynx. So air would flow in this manner, going from the nose all the way back and then down the throat. It would then reach a forked path where it could either go down the esophagus or the larynx. Now, air is obviously not going to go down the esophagus. Instead, it will head down the larynx. Now, the upper respiratory system pathway, air is going to flow through the nose, inferior, middle, and superior nasal concave, those ridges that actually make the air more turbulent, swirl it around to help get debris stuck in there, as well as warm and moisten the air. It is going to, of course, be involved in scent to get some of that air into the olfactory epithelium to detect odor, and then will extend its way back to the nasopharynx area. Now that we've worked our way through the actual nasal and oral cavities, we're bringing air back to the pharynx. The pharynx is quite simply the back of the throat. It's a passageway that connects the nose to the mouth and the back of the throat. The nasopharynx is the back of the nose area. It opens to the escutcheon or auditory tube and has the pharyngeal tonsils in it. The oropharynx, the next one down, is going to be the back of the mouth area. This actually has a structure called the uvula in it. The uvula is a cone-like soft tissue projection from the soft palate that contributes to human vocalizations. It's very common in Germanic, African, click languages, and others to where the uvula is used a little bit more. And this helps to also block the internal nares during swallowing to prevent nasal regurgitation. Interestingly, some people have their uvula pierced. Not sure why. It's just something that people do. The palatine tonsils are on either side of the uvula, and they are sometimes removed as children, usually from strep throat, tonsillitis, things like that. The laryngopharynx is going to be the next one down. This is the area that has the entrance to the trachea and then down into the esophagus, so where it actually divides into these pathways. Here again, we can see the actual pathways of the respiratory tract. We've already talked about, again, there's going to be the nasopharynx here, the oropharynx back here, and the laryngopharynx here. From the laryngopharynx, we're going to work our way down the larynx. Working your way down into the larynx, first we can see the uvula, and then we can see the different actual tonsils on either side. Now that we've inhaled air and made it leave the pharynx by passing through a narrow region called the glottis, we're going to enter into a structure called the larynx. The larynx is a complex structure that involves several muscles, ligaments, and cartilages. It functions to ensure that air moves down the trachea and food moves down the esophagus. It helps with the act of swallowing, and it contributes greatly to the production of human vocalization. Some of the structures associated with the larynx first are going to be the thyroid cartilage, which contains the laryngeal prominence, more commonly known as the Adam's apple. This is composed, of course, of hyaline cartilage and forms a shield on the anterior and lateral walls of the larynx. Cricoid cartilage is going to be the next one. Cricoid cartilage is simply going to be another band of hyaline cartilage that will sit lower and act as an anchor or base for the thyroid cartilage. on the backside of the thyroid cartilage is going to be the epiglottis. This is elastic cartilage that will actually project over the glottis and is connected to the thyroid cartilage and the hyoid bone. During swallowing, elevation of the larynx and hyoid causes the epiglottis to fold back onto the glottis. So this prevents aspiration of foods and liquids into the trachea, aka prevents stuff from going down the wrong pipe. Here we can actually see a diagram of the larynx. Note the thyroid cartilage, large hyaline cartilage shield with the laryngeal prominence sitting at its center. On top of the thyroid cartilage normally is going to be where the thyroid gland would actually sit. Just below the thyroid cartilage, or inferior to it, is going to be the cricoid cartilage. This is the base or anchor that's going to be connected by different cricothyroid ligaments throughout. Superior to the thyroid cartilage is actually the hyoid bone, and this will be connected by a number of ligaments as well as different muscle structures. Then on the posterior aspect, we would have the epiglottis. Here on the posterior aspect where we can see the epiglottis, it's going to connect itself down via several ligaments to the arytenoid cartilage and to the thyroid cartilage. Arytenoid cartilage is going to be two triangular shaped hyaline cartilage ridges that would actually project over the glottis itself. Now, glottis itself is not a structure directly, but more of an opening. Here we can see the glottis and the vocal cords. Now, the vocal cords themselves are delicate, elastic vocal folds. These are the true vocal cords. They lie in the glottis medial to the inelastic vestibular folds, or your false vocal cords. The vestibular folds provide protection for the vocal folds and help to prevent foreign objects from entering the glottis. Again, the glottis is simply going to be the hole or opening that would lead itself down to the respiratory tract. Now, during swallowing, we have an interesting thing that happens. So we swallow a bolus of food. As it pushes its way down, the action of swallowing is going to move the hyoid bone up and back, forcing food back down the esophagus and forcing the epiglottis closed over the larynx. This, of course, is going to help to prevent any sort of aspiration or taking food down your windpipe. From the larynx, we now extend ourselves down the full windpipe known as the trachea. The trachea itself is going to be composed of several tracheal cartilage rings. These rings are actually C-shaped and not a complete ring. This is designed so that way the diameter of the actual trachea can increase or decrease in size. The lining of the trachea is going to be composed of a respiratory epithelial mucosa, the pseudostratified that we've already talked about, and the posterior side of the cartilage ring is going to consist of the trachealis muscle. This is the muscle that will actually allow those C-shaped cartilage rings to constrict or dilate based upon the actions of the body and the necessities that it requires. Here we can see a nice diagram of the trachea as it branches down from the larynx, and then it will bifurcate into the primary bronchi. This is actually going to be a look inside the trachea. I want you to notice the C-shaped structure of the rings and then the smooth posterior aspect of the trachea where they are not connected. This, of course, is going to be the trachealis muscle that would allow itself to contract, bringing the rings in, thereby constricting the actual trachea, or it would relax, allowing the C-shaped rings to open up, thereby allowing for dilation. Here's a histological view where we can see it even more. We can also see the relationship between the trachea and the esophagus. The trachea, again, is going to have the C-shaped rings that would actually surround the lumen. The posterior aspect is going to have the trachealis muscle, and just behind this is going to be the esophagus. The trachea is going to end when it starts to split. It will split into the left and right primary bronchi. The primary bronchi is steeper and larger in diameter than the left. Each primary bronchus will enter the lung at the point called the hilum. Hilum is simply the point of entrance for the bronchi as well as the arteries and veins. The combination of the bronchus, artery, and vein is called the root. Here we can see the steeper angle of the right lung where it comes off and branches down. And then we have, of course, the bronchus of the left lung where it extends out. Now the lungs themselves are going to have the apex that points superior, so it's going to be at the top of the lung, and the base that's inferior. To simply remember this, remember the apex is the top, the base is the bottom of the lung. Now the right lung has 3 lobes, a superior, middle, and inferior lobe, whereas the left lung has 2 lobes. It only has a superior and an inferior lobe. It also contains the oblique fissure that would divide these 2 lobes, and then a very unique structure, shape, that would be called the cardiac notch. This, of course, is because the heart is going to sit off to the left side of the chest, and thereby causing the left lung to have to grow and adapt around it. Here we can see the shape of the different lungs, the right and the left lung. We can notice the apex is at the top and the base is at the bottom. Take note of the right lung first, with its superior, middle, and inferior lobes. It's divided by a horizontal fissure and the oblique fissure. The left lung is going to have just the superior and inferior lobes and be divided by an oblique fissure, and also feature the cardiac notch in the superior lobe. This is again to allow the heart to sit off to the left side. This is an internal view on the inside of both the right and the left lung to simply show you where the hilum is and where the root would be. This is going to be where the left and right bronchus are going to insert, as well as the pulmonary arteries and veins would insert directly into the lungs. If we remember from way back during our first lecture series in Biology 177, we talked about one of the serous membranes. That serous membrane in this case is the pleural membrane. Remember that the pleura is going to line each lung with a separate serous cavity consisting of a visceral and a parietal pleura. The visceral pleura is going to actually touch the lung. Remember, visceral, organ, touching the actual organ itself. The parietal is going to line the inside of the thoracic wall and have space between them. The space that is created between the visceral and parietal is the pleural cavity, and this is going to, of course, contain the pleural fluid. Here we can actually see a transverse view of the lungs, and we can see the space in between the actual lung and the thoracic wall. Now that we've worked our way inside of the lung, from the hilum, and then past the visceral pleura, we can start to branch off from there. Here we can see the different branchings of the actual bronchi and how they get a little bit smaller as we work our way down. Every time that the bronchi splits, it's going to become a new structure. In this case, the primary bronchus is actually going to become, the primary bronchus that we can see here, is going to split and become the secondary bronchus that we see here. From there, it will then split again into the tertiary bronchi that we can see here. The tertiary bronchi are going to have, and above would have cartilage plates. This is to make sure that it maintains its shape during breathing, because otherwise it's not strong enough to actually support itself. It's going to need these extra cartilage rings in order to create structural support. But from the tertiary bronchi, we get to the bronchioles down here. Now the bronchioles themselves are actually not going to have cartilage directly. The subsequent branching of the tertiary bronchi leads through many divisions to a large number of terminal bronchioles. Each terminal bronchiole is going to have considerable smooth muscle in the walls, and this determines the resistance to airflow. During an asthmatic attack, smooth muscle in these terminal bronchioles bronchoconstrict, cutting off airflow to the alveoli. Each terminal bronchiole divides into several respiratory bronchioles, as can be seen here. So we go bronchioles to terminal bronchioles to respiratory bronchioles, and then to the alveoli themselves, which would be inside the lobule. So the bronchioles are going to have the tertiary bronchi that give rise to these. They are self-supporting and they do not require cartilage plates. They consist of smooth muscle, as already discussed, that allows for bronchodilation and bronchoconstriction. The bronchioles will terminate with clusters of alveolar sacs, or lobules. Once we reach to the alveolar ducts and alveoli, we've reached the actual site of gas exchange. Now, each lung has about 150 million alveoli, and there's an extensive capillary network that surrounds each alveolus. The capillaries here are going to drop off carbon dioxide and pick up oxygen. And there's elastic tissue around each alveolus to allow it to maintain shape and position during inhalation and exhalation. Here we can see a picture of the alveolar sacs as they connect to the terminal bronchioles and then to the respiratory bronchioles. They look like a bunch of grapes surrounded by a netting of capillaries. This increased kind of circular structure is going to allow for an increase of surface area for a greater site of gas exchange. Now, inside the alveoli, we have some very specific structures. The lining of the alveoli is going to consist of simple squamous epithelial cells. It is going to have these broken up into two types. The first one is called type 1 alveolar cell, and the second one is called type 2. Now, type 1 alveolar cells are going to be involved in gas exchange. This is going to be the one that actually exchanges the gases, the oxygen and carbon dioxide. Type 2 alveolar cells are going to be scattered amongst the type 1, and they're actually going to make a substance called surfactant. Surfactant, its main function is to prevent alveolar collapse. What it is overall is going to be kind of a soapy lipoprotein. Now, soap what it actually does to water is going to reduce the surface tension. This makes water wetter, is the best way to think about it. It makes it more slippery so that things won't stick to each other. Namely, if the lung is going to, during exhalation, collapse down, the lungs, the alveoli, won't stick to each other. They'll be able to glide off of each other very nicely. Here we can see some of the structures of the internal alveolus, including the capillaries that would surround the different alveolar sacs, the type 1 cells, which are going to be in the white, and then we can see some of the type 2 cells that are going to be on the alveolar wall in the green. And the type 2 are the ones that secrete surfactant. Okay. Now, gas exchange that occurs at the alveoli. Most of the lung tissue in the place where gas exchange actually occurs is comprised of small air-filled sacs called alveoli that are highly specialized for the efficient transfer of gases between lungs and blood. Now, the alveoli themselves are going to be a single layer of thin epithelium lying on a very thin basement membrane, as can be seen in the respiratory membrane picture where it's fused together. Respiratory capillaries cover up to 90% of the alveolar surface to maximize the exchange surface. Now, these capillaries, like I said, are physically fused to the alveolar cells, specifically the type 1, to facilitate gas exchange. This helps to reduce diffusion distance, thereby increasing rate of diffusion. And the type 2 alveolar cells that exist throughout, but much fewer quantity than the type 1, are going to be a little thicker and function to secrete surfactant. And surfactant, again, is going to help make sure that the lungs do not collapse. Now that we've finished with the anatomy of the respiratory system, we're going to talk about the laws that actually govern gases. Gases are measured in terms of pressure. This is how much of a gas there is in a specific space and the amount of pressure that it exerts on its environment. In many respects, the movement of gases is much like the movement of fluids. The air we breathe has a pressure associated with it, and we call this atmospheric pressure. It is the product of the sum weight of all gases in the atmosphere, and it extends up to the outer reaches of our global atmosphere. Now, at sea level, our average pressure is 760. The first law that we're going to actually look at is called Dalton's law. Dalton's law quite simply states that the total pressure in a gas mixture is the sum of the partial pressures of each individual gas. This means that our atmospheric pressure is the sum of all the gases that are inside of it. To give you more of an example of Dalton's law, we'll look at first the gases that make up our atmosphere. Those would be nitrogen, oxygen, and carbon dioxide. There are, of course, a few other trace gases, but they're not in a high enough concentration to really be super relevant to this discussion. Nitrogen is going to make up 78%, oxygen 21%, and carbon dioxide 0.04%. Now remember what I said before, the pressure of the atmosphere at sea level is 760 millimeters of mercury. This means that when we take 78% of 760, we get 593 millimeters of mercury. And then same thing for oxygen and CO2. Oxygen is 160 millimeters of mercury, and CO2 is about 0.30. Now these numbers have been rounded a little bit, but we're going to use them for the sake of this example. So the sum of all three of these is going to equal the pressure of the atmosphere at sea level. Now obviously this is going to change over time because there's different altitudes throughout. So as the altitude increases, the pressure is going to go down. So that means these numbers would change. Consequently, if we want to actually introduce humidified air, we're going to have to deal with water vapor. To calculate with humidity, we simply subtract the pressure of the water vapor from the atmospheric pressure before taking percentages of the gas. Now, for this example, fully humidified air pressure is going to be 47 millimeters of mercury. So we would take that 47 millimeters of mercury and subtract from the 760 to get 713. So then we take that 713 and we are now looking at the percentages of each one of those. So 78% of 713 is going to be 556 millimeters of mercury for nitrogen. And the same goes for oxygen and CO2 with their respective percentages. Notice that it's actually gone down so that we have less oxygen in the atmosphere that we can breathe when the air is humidified as opposed to when it is not humidified. This is why when there's a lot of humidity in the air, it suddenly becomes a little bit more difficult to breathe, or breathing is heavier. The other major law that we need to concern ourselves with is Boyle's law. Boyle's law looks at a pressure-volume relationship. In a closed container, pressure is created by the collision of gas particles against the walls of the container. If you reduce the size of the container, the collisions will increase, and therefore so does pressure. Another way of saying this is quite simply that pressure and volume are inversely related. And this is shown by many of the gas laws that are out there. What this means is that if you increase pressure on a gas, you compress it and reduce its volume. This is different than for fluids, which are incompressible. If you begin with a container under a certain gas pressure and with a certain volume, and then you compress it, the volume will decrease accordingly. Now this is summarized by another formula called pressure 1, volume 1 equals pressure 2, volume 2. This relationship between pressure and volume explains how the lungs work, and we're going to look at that in the next lecture set. Now when we're talking about gas flow dynamics, airflow in the respiratory tract follows the same laws that blood and fluid flow follows in the cardiovascular system. Remembering that flow is proportional to a difference in pressure and airflow velocity, and volume and flow volume decreases as resistance increases is really going to help you understand how the air flows through the system. And it's really the same as blood. And we'll talk about that more in the next lecture.